The next speaker is Anna Teles. I'm going to promote the panelist.
Thanks a lot for this opportunity. I really appreciate it. And today I would like to speak about our work on the modulation of naive T-cell repertoire diversity by the individual MHC2 in our model, that is the stickleback fish. And just to start, we all know that the evolution has favored the balance in immunity, so the organisms should be able to recognize all the pathogens that can be harmful, and at the same time be able to protect the self. So this recognition of the different antigens is mainly modulated by the adaptive immune system, and in the center of this adaptive immune system we have T cells, and T cells they come from the bone marrow to the thymus, where they go to a process of maturation. During this maturation, they acquire the T cell repertoire receptors that can recognize peptides presented by the MHC in the surface of epithelial cells, thymic epithelial cells. And first they go into a process of positive selection, where there is the deletion of T cells that they do not interact with the peptide MHC molecules, and only the ones that interact can survive, and then go to a process of negative selection, in which the cells that bind with a very high affinity with the MHC peptides, they go through clonal deletion, and only the ones that have an intermediary to low affinity, they can pass to the periphery. This allows the immune system to differentiate, to be able to protect against autoimmunity. In the periphery, then the cells can encounter the peptides presented by the MHC of antigen-presenting cells, and then a higher diversity of the receptors of T cells would allow the recognition of a higher number of pathogens, and this would be good for the host. Because in the thymus we have the two different processes, the positive and negative selection, a higher MHC diversity in the thymus would allow a higher diversity of T cells, because there is a chance that these T cells recognize the peptides and bind and pass to the second step. But negative selection, having a higher diversity, would in this step would lead to the deletion of T cells, because they would also have higher chances of binding with a strong affinity to the MHC peptide. Therefore, individuals with a too high MHC diversity must have a depleted TCR repertoire due to the deletion of these T cells that react with self-MHC peptides combinations during development. And this is what we propose to test. This was already proposed in the 90s in a theoretical paper that was proposing that the optimal number of MHC complex molecules in individual is an intermediary and not higher or low. And we proposed to study this in stickleback fish. We choose this model because these are very small organisms that lives in a very wide variety of habitats in the North Hemisphere. It exists from the sea to the lakes to the rivers, so it can be studied in different settings. And for the ones who don't know, this fish is a very small fish, like this size. And this is a male that is teasing the female inside the nest. So the males are the ones that build the nest, and the females just go inside and lay the eggs, and they take care of, the males take care of the eggs. And these fish are still a good model for studying adaptive immunity, because they have a completely functional adaptive immune system with a natural level of MHC2 diversity that is comparable to the humans. So they are suitable for these studies. And what was already shown is that the frequency of MHC alleles in the population of stickleback fish in different lakes, for example, has already an intermediary number of MHC alleles. So it's not, the frequency is higher when the number of MHC alleles is intermediary. And this is also demonstrated, so this selection is also demonstrated by the number of parasite load. So was also shown to be low when there is an intermediary number of MHC. In our setting, we propose to look for the naive T cell repertoire of stickleback fish with different MHC, with different number of MHC alleles that can go for very low, 1 allele only, to 7 alleles. And then we sequence and analyze the TCR genes from the whole body because they are very small, so we can take the whole organism. And these individuals are only 2.5 months old, which is really naive. And first we had to characterize the VD and J genes of the T cell receptors, and we found that it's very diverse, and we have both TR beta 1 and TR beta 2 like in other fish. And then we look for the sequences of the T cells of these naive fish, and we found like that most of the cells only exist once. So most of the receptor, the CDR3 that we found, we only found it in one cell. Most were like singletons, and some 7% was showing in doubletons. And this was similar also to what is found in mice and humans. For instance, that the number of singletons is much higher when the individuals are young compared when they are old. So this just shows that it's a naive cell repertoire. And then when we were looking, when we were comparing the number of alleles with the number of CDR3 clonotypes, we found that the diversity of the T cells, so the number of CDR3 clonotypes, was higher when there was a low to intermediate number of MHC alleles, and not when it was a higher number of MHC alleles. And the same could be seen to the level of the family. So individuals with a lower number of a median number of individual MHC alleles have a higher diversity of the CDR3 than individuals with a higher median individual number of MHC alleles. And also, very interestingly, we found that charge, hydropathy, and strength of the central CDR3 amino acids was higher in individuals with a low to intermediary number of MHC alleles. And this just suggests that there are regulatory mechanisms that are mediated by the MHC during the selection of T cells. So to summarize, most of the T cells seem to be naive in the young fish, which was already expected. And the TCR diversity is significantly higher in individuals with low to intermediary number of MHC alleles. And also the number and diversity of these MHCs influence the CDR3 amino acid properties. So we had some data that enforce this theory of MHC TCR coevolution that says that it's an optimized, not a maximized number of MHC molecules in individuals that favor an optimal diversity of T cells. So for last, I would like to acknowledge the people that work with me here, mainly Tobias Lenz and Manfred, the Max Planck Institute of Evolutionary Biology, and also to our collaborators, Thomas Bohm. And thank you for listening.
Thank you, Anna. I have a question. The cases with the few alleles, let's say 2 or 3 alleles, they have always the same 2 or 3 alleles, or is a mixed population that you have a different combination of alleles? among your population?
Yeah, it's different. So we have many different alleles. Even one is just one allele, so just one that has one. But the 2 and 3, it's a mix. So we have different allele types.
Combination. Okay. Thank you. If there are no questions, okay, there is one coming from Jamie. So is about 6 the golden number for all species that have TCR MHC base immunity, or does it vary?
Didn't understand.
Okay, is about 6 the golden number of all species that have TCR MHC base immunity, or does it vary?
6?
So you show that with 2 or 3, you have more diversity, I think, in your model.
Yeah, 2 and 3 is higher, 6 and 7 is lower. So yeah, with the lower intermediate, the diversity is higher. So probably about the MHC, no? The MHC.
So there is a follow-up. Is from the
MHC, right?
Yes, still from Jamie. Is the optimal number shared across species, maybe? Okay. Oh yeah. Okay. Number of alleles.
Yeah. This typing was different from ours, but here, so yeah, these are fish from Canada, and they have a higher number of MHC too. Yeah. But here was 6.
It might vary between species.
Yeah, exactly.
Okay. So we have a question from Incarnita. Have you looked at the overlap between fish with different MHC alleles?
Yes, was very low overlap. And this is something that we want to increase the number of species that we sequence to see if it's really like this or if it's just with the sequencing number of cells.
Okay. If there are no more questions, we can move. We are ahead of time. Oh, thank you. Ankarnita is answering. Thank you. And we can move to the software demonstration. And Victor should moderate the session. Thank you, Anna.
Thank you.